Hello everyone, Professor Beda here. Let's discuss how cells regulate gene expression. Prokaryotes and eukaryotes precisely regulate gene expression in response to environmental conditions. In a multicellular eukaryote, genes expression regulates development and is responsible for differences in cell types. Many genes of the bacterial genome are switched on or off by changes in the metabolic status of the cell. Some genes are regulated singly, and others as groups of related genes. One basic mechanism for this type of regulation of groups of genes in bacteria describes as the operon model. A key advantages of grouping genes of related function into one transcription unit is that single on-off switch can control the whole cluster of functionally related genes. In other words, these genes are coordinately controlled. The on-off switch is a segment of DNA called an operator. Both its location and name suit its function. Positions within the promoter or in some cases between the promoter and the enzyme coding genes, the operator control the access of RNA polymerase to the genes. Together, the operator, the promoter, and the genes they control the entire stretch of DNA required for a protein production pathway constitute an operon. Um, if the operator is the operons switch from controlling transcription, how does this switch work? It works by itself. The operon is turned on. That is, RNA polymerase can bind to the promoter and transcribe the gene of the operon. The operon can be switched off by a protein that is called a repressor, binds to the operator, preventing RNA polymerase from transcribing the genes, often by preventing RNA polymerase from binding. A repressor protein is a specific for the operator of a particular operand. For example, the tryptophan repressor, which switched off the tryptophan operon by binding to the tryptophan operator, has no effect on other operons in the E. coli genome. A repressor protein is encoded by regulatory gene. All organisms, whether prokaryotes or eukaryotes, must regulate which genes are expressed at any given time. Both unicellular organisms and the cells of multicellular organisms continually turns genes on and off in response to signals from their external and internal environment. Regulation of gene expression is also essential for cell specialization in multicellular organisms, which are made up of different types of cells. To perform its own distinct role, each cell type must maintain a specific program of gene expression, in which certain genes are expressed and others are not. A typical human cell might express about a third to half of its protein coding genes at any given time. Almost all cells in multicellular organisms contain an identical genome. A subset of genes is expressed in each cell type. Some of these, about 35%, are housekeeping genes expressed by many cell types, while others are unique to that cell type. The unequally expressed genes allow these cells to carry out their specific function. The differences between cell types, therefore, are due not to different genes being present, but to differential gene expression. The expression of different genes by cells with the same genome. The function of any cell depends on its expressing the appropriate set of genes. The transcription factors of a cell must be locate the right genes at a right time. A task like finding a needle in a haystack, uh, abnormal genome expression can cause serious imbalances and disease, including cancer. Genes are regulated at many stages. In nucleus, the first stage in chromatin modification, the second in transcription and mRNA processing. In cytoplasm, translation, mRNA degradation, and protein processing degradation. We will discuss the only transcription uh, regulation of gene expression only about the transcription. Eukaryotic gene and the DNA elements or segments that control it are typically organized. We call that a cluster of protein called a transcription initiation complex symbol on the promoter sequence at the upstream end of the gene. One of these proteins, RNA polymerase 2, then proceed to transcribe the gene, synthesizing a primary RNA transcript pre-mRNA. RNA processing includes enzymatic addition of 5' cap and a poly A tail, as well as splicing out of introns to yield a mature mRNA. Associated with most eukaryotic genes are multiple control elements, segments of non-coding DNA that serve as binding sites for the protein called transcription factors, which bind to the control elements and regulate transcription. Control elements on the DNA and the transcription factors that bind to them are critical to the precise regulation of gene expression seen in different subtypes. In eukaryotes, the rate of gene expression can be strongly increased or decreased by the binding of specific transcription factors, either activator or repressors, to the control elements of enhancers. Researchers have identified two types of structural domains that are commonly found in a large number of transcription activators: a DNA binding domain, a part of the protein's three-dimensional structure that binds to DNA, and one or more activation domain. Activation domains bind other regulatory proteins or components of the transcription machinery, facilitating a series of protein-protein interactions that result in enhanced transcription of a given genome. In this model, for the action of enhancers and the transcription activators, binding of the DNA by protein enables enhancers to influence a promoter hundreds or even thousands of nucleotides away. Specific transcription factors activators bind to the enhancer DNA sequences and then to a group of mediator proteins. These in turn bind to general transcription factors and then RNA polymerase 2, thus assembling the transcription initiation complex. These protein-protein interactions lead to correct positioning of the complex on the promoter and the initiation of RNA synthesis. Only one enhancer with three gold control elements is shown here, but a gene may have several enhancers that act at different times or in different subtypes. Activator proteins binds to distal control elements. grouped as enhancer in the DNA. This enhancer has three binding sites, each called distal control element. A DNA binding protein brings the um, bond activators closer to a promoter. General transcription factor, mediator protein, and RNA polymerase 2 are nearby. Uh, the activators bind to a certain mediator protein and general transcription factor, helping them form an active transcription initiation complex on the promoter. In addition to influencing transcription directly, some activator and repressors indirectly affect chromatin structure. Some repressors recruit proteins that remove acetyl group from histones, leading to reduced transcription, a phenomenon referred to as splicing. Indeed, recruitment of chromatin-modifying proteins seem to be the most common mechanism of repressing in eukaryotic cells. In eukaryotes, the precise control of transcription depends largely on the binding of activators to DNA control elements. Considering that many genes must be regulated in a typical animal or plant cell, the number of different nucleotide sequences in control elements is surprisingly small. A dozen or so short nucleotide sequences appear again and again in the control element for different genes. On average, each enhancer is composed of about 10 control elements, each binding only one or two specific transcription factors. It is the particular combination of control elements and enhancer associated with a gene, rather than a single unique control element that is important in regulating transcription of the gene. Even with only a dozen control element sequences available, many combinations are possible. Each combination of control elements can activate transcription only when the appropriate transcription activators are present, which may occur at precise time during development or in a particular cell type. This figure illustrates how the use of different combination of just a few control elements can allow differential regulation of transcription into representative cell types, liver cells and lens cells. This can occur because each cell type contains a different group of transcription activators. Both liver cells and lens cells have the genes for making the protein albumin and crystalline, but only liver cells make albumin. Uh, this is a blood protein, and only lens cells make crystalline, the main protein for the lens of the eye. The specific transcription factors made in a cell determine which gene are expressed. Um, in this example, the genes for albumin and crystalline are shown at the top, each with an enhancer made up of three different control elements. Although the enhancers for the two genes both have a gray control element, each enhancer has a unique combination of elements. All activator proteins required for high-level expression of the albumin gene are present in liver cells only on the left, whereas the activators needed for expression of the crystalline gene are present in the lens cells only in the left and the right. Sorry. For simplicity, at your textbook, consider only the role of specific transcription factors that are activators here. Ortho repressors may also influence transcription in certain cell types. So here the DNA in liver showing available activators. The albumin gene is expressed, but the crystalline gene is not. On the right, DNA in the cell showing available activators. The crystalline gene is expressed, but albumin gene is not.